Okay, so welcome back to this next video in which we are discussing the cell cycle. Okay, right, so we're currently discussing the pathway downstream of growth factor binding to a growth factor receptor. Okay, so we've seen that we get the activation of this family of monomeric G proteins known as Ras monomeric G proteins. Okay, and these family members all seem to pretty much do the same thing. They lead to the activation of RAF kinases, okay? And this seems to occur via the phosphorylation of a tyrosine residue on the RAF kinase. And it's still a little bit of a mystery as to how this actually occurs, but it's definitely through interaction with the Ras G protein. Okay, now, RAF kinases then are serine-threonine kinases, so I want to just explain to you what that means. Okay, so let me show you the structure of a serine residue and a threonine residue. We've talked about tyrosine phosphorylation, but we haven't talked about serine and threonine phosphorylation, so I want to talk about this. So serine and threonine are other amino acids which you can have within proteins. Okay, so I'll draw once again their residue structure. So here's the amino group, here's the alpha carbon with a hydrogen coming off it, and here's the carboxylic acid group. And again, I've drawn it as though it's within a polypeptide. So I've drawn the amino group as though it's bound to the carboxylic acid group of the amino acid in front of it, and I've drawn the carboxylic acid group as though it's bound to the amino group of the amino acid behind it. Okay, and the R group then of a serine residue consists of a methylene group with an alcohol group coming off it. Okay, so this is the structure of a serine residue. Okay, meanwhile, if I show you the structure of a threonine residue, it's very similar to a serine residue. So once again, here is our core amino acid structure, an amino group, an alpha carbon with a hydrogen coming off it, a carboxylic acid group, and then the structure of a threonine residue contains a single carbon again coming off, okay, with a hydrogen and an alcohol group, but then instead of having a second hydrogen coming off this carbon, you have a methyl group here. Okay, so this here, this methyl group is what distinguishes threonine from serine then. Okay, so this amino acid here, this is threonine. Right, so both serine and threonine residues can be phosphorylated, similar to the way that tyrosine can be phosphorylated. So once again, I'll show you the principle of this. Again, serine-threonine kinase enzymes, they don't use pure inorganic phosphate groups to actually attach onto serine and threonine residues. Instead, they take the phosphate group from an ATP molecule and use the energy released from the hydrolysis of ATP to bind the phosphate group onto the alcohol groups of the serine or threonine residues. Okay, but I will show you this as though we're attaching on an inorganic phosphate because it gets the principle across, even though it's not actually what's going to be happening. Okay, right, so here once again is our phosphate group, and you can imagine that this is exactly the same for threonine here. We've just got that extra methyl group. And what then again will happen is you'll form this ester-like link known as a phosphoester link. So you'll take the alcohol group of the phosphate group, you'll take a hydrogen off the alcohol group of the serine or threonine residue, and you'll then bind the oxygen of the serine or threonine residue to the phosphorus atom of the phosphate group. Okay, and this link that we've now formed here between the oxygen atom and the phosphorus atom, this is known as a phosphoester link. Okay, so we're going to attach phosphate groups onto serine and threonine residues via phosphoester links. And this is what it means to phosphorylate serine and threonine residues. So serine-threonine kinases, okay, often abbreviated to SIR, which is the three-letter abbreviation for serine, THR, which is the three-letter abbreviation for threonine, and then kinase. What these are going to do is they're going to phosphorylate serine and threonine residues within proteins, and this is going to moderate the function of those proteins. Okay, or modulate the function of those proteins. So we have just got the activation of this RAF kinase. Okay, so here is our RAF kinase, and remember there were those three types of RAF enzyme. ARAF, BRAF, and CRAF in humans. Okay, but they all pretty much do the same thing, so we're not going to distinguish between them. And we've now got this phosphate group that's off a tyrosine residue within this RAF enzyme here. Okay, so I'll colour in this RAF enzyme in red. Now, what's going to happen then is that this RAF enzyme is now going to phosphorylate another protein kinase enzyme. Okay, and this is why these sort of cascades are often called kinase cascades. You'll often see them referred to something like this, because effectively what you're going to get is the activation of one kinase enzyme is going to phosphorylate another kinase enzyme, activating it, which will then phosphorylate another one. Okay, so we'll see this happen. So our activated RAF here is going to work on enzymes known as MEK. Okay, so here we'll have our next enzyme along, which is known as MEK. Now, what does MEK stand for? MEK stands for MAPK/ERK kinase. Okay, and I'll write that down for you. So the M is for MAPK, which is the name of the enzyme that this enzyme here is going to itself activate. Okay, and we'll see that enzyme in a moment. Now, this enzyme also has another name. It's also known as ERK. Okay, and we'll see what both of these stand for in a moment. So the E then here is for ERK. Okay, and then it's kinase. So because the target of this enzyme has two names, MAPK and ERK, we say that this enzyme can be called the MAPK/ERK kinase. Okay, right, so let's colour this one in yellow. Now, again, there is not just one form of MAPK/ERK kinase. Okay, there are actually two forms of it. There is MEK1 and MEK2, but again, they do pretty much the same thing, so we won't distinguish between the two different forms of this enzyme. Right, so here comes along our RAF kinase enzyme, which is now activated. It's going to phosphorylate serine and threonine residues within our MEK protein kinase enzyme here. So you're going to get phosphorylations occurring to your MEK enzyme here. And this now is going to activate our MEK enzyme. So we've activated this RAF enzyme so that it can then activate another kinase enzyme. Now, MEK is a little bit more complicated. It can phosphorylate pretty much whatever it likes. It can phosphorylate serine residues, it can phosphorylate threonine residues, it can also phosphorylate tyrosine residues. So it's a quite broad kinase. It's rare that you find kinases which can phosphorylate all three types of residues, serine, threonine, and tyrosine, but MEK can do that. Okay, and it's now going to phosphorylate residues within the next target along, which is another kinase enzyme. So let's introduce our final family of, the final kinase enzyme in this pathway. Okay, which is finally MAPK. Okay, or you can call it ERK. Okay, so now what do these two names stand for? Well, MAPK first. This stands for mitogen activated protein kinase. Okay, activated protein kinase. So the M is for mitogen, the A is for activated, the P is for protein, and the K is then for kinase. So this is another kinase enzyme. Now, for short, the mitogen activated protein kinase is often abbreviated to MAPK, or more commonly you'll hear people refer to this as MAP kinase. So they'll abbreviate the mitogen activated protein bit down to MAP, and then they'll just speak the kinase, so they'll call it MAP kinase. Okay, that's the most common thing to hear this enzyme referred to as. Now, the other name that is used for these enzymes is ERK. Okay, and what does ERK stand for? ERK stands for extracellular, that's the E, and then there is also a letter that doesn't appear in the name. Okay, and this is signal, so extracellular signal. And then the R is for regulated, so this is the extracellular signal regulated, and then of course the K is for kinase. So ERK stands for extracellular signal regulated kinase. Okay, so the three important ones are extracellular, regulated, and kinase, and that's the ERK naming here. Okay, right. Now again, there are two different types of MAPK ERK enzymes. Okay, and these are known as ERK1 and ERK2. Okay, so there's the extracellular signal regulated kinase 1 and the extracellular signal regulated kinase 2. Okay, and again, for our purposes, these do pretty much exactly the same thing, so we won't distinguish between them. We'll just say we're dealing with an mitogen-activated protein kinase or an extracellular signal regulated kinase or a MAP kinase or an ERK. Okay, so I'll use the abbreviation ERK then. And what's now going to happen is that the MEK enzymes, which are the mitogen-activated protein kinase / extracellular signal-regulated kinase, because they're going to phosphorylate these enzymes here, they're going to phosphorylate these enzymes and they're going to activate them. Okay, so ERK enzymes get phosphorylated by MEK enzymes and they themselves become activated. Now, it is for this reason that sometimes you will hear the MEK enzymes referred to as the mitogen-activated protein kinase. Okay, so some people will call the MEK enzymes MAPKK. Okay, because they are the kinase enzymes for the mitogen-activated protein kinases. So you put an extra K there. Now, this gets even more ridiculous because some people will call the RAF kinase enzymes the mitogen-activated protein kinase, because these are the kinase enzymes for the mitogen-activated protein kinase. So then it gets even more ridiculous down here. And some people will also write these names as MAP3K, okay, to denote the kinase. They'll call this one also MAP2K. So those are other names. And the thing that makes this pathway the most, as complicated as it is because there are just a plethora of different names for the same proteins, okay? That's the nightmare of this pathway. Okay, so these are other names that you can use for these enzymes, but I would stick to RAF, MEK, and ERK, or you do hear ERK referred to as MAP kinase, so be aware of that one, but don't worry about these mitogen-activated protein kinase names, but you will see them occasionally. Okay, right, so all of this then has come down to the activation of our ERK kinase enzyme. Now what's going to happen is that our ERK kinase enzyme is going to go into the nucleus of our cell and it's going to activate transcription factors. Okay, so it's going to phosphorylate key transcription factors and change their activity. So in it comes to the nucleus. Okay, so all of this kinase cascade, as it's called, and now you can appreciate this term. It's a cascade of kinases activating other kinases. All of this was occurring in the cytoplasm. Now, finally, the activated mitogen-activated protein kinase, or ERK enzyme, is going to come into the nucleus, and in the nucleus, it's now going to phosphorylate and activate certain transcription factors. Okay, so which two transcription factors is it going to phosphorylate? Well, one of them is known as ELK1. Okay, so let's have an ELK1 transcription factor here. Okay, so I'll colour in ELK1 in yellow. So at the moment, ELK1 is sitting nice and inactive, and now it's going to get phosphorylated by our mitogen-activated protein kinase. Okay, so here we go. And now what ELK1 is going to do is it's going to act as a transcription factor. Okay, so I just need to explain to you the concept of a transcription factor. So I'll draw these two lines here, and now these two lines do not represent a cell membrane anymore. They don't represent the inner and outer leaf, but instead I'm going to use these two lines to represent a piece of double-stranded DNA. Okay, so we've got two strands of DNA that are complementary to one another. And let's say we have a gene here. So, what's the concept then of a transcription factor? Well, upstream of all genes in eukaryotic cells, okay, so this portion that I'm outlining in green, this is representing a gene. Upstream of all genes in eukaryotic cells, you have a special region which is known as a promoter region. Okay, so this portion in pink here, this is the promoter region. Okay, now the promoter region is not involved in being translated into protein itself. Okay, so it will not be read by ribosome and turned into a sequence of amino acids. However, it's extremely important in determining how much of the gene product of the downstream gene you are actually going to produce. And the reason it does this is because it's the place where the RNA polymerase II enzyme binds to the DNA, opens the DNA, and works its way along to produce a piece of mRNA. So to say that again, what happens is if you want to make the gene product of this gene, RNA polymerase II comes in, binds to the promoter region, works its way along the DNA, synthesizes a piece of mRNA that is complementary to the coding strand. That mRNA will then go through post-transcriptional modifications, okay, then will go out into the cytoplasm to be translated into protein, basically. Okay, so if the promoter region has a very high affinity for binding to RNA polymerase II, then RNA polymerase II will bind there all the time. Okay, you'll get mRNA being produced all the time, and therefore you'll produce a large amount of protein. If the promoter region has a very low affinity for binding to RNA polymerase II, then RNA polymerase II will hardly ever bind to that promoter region. Okay, you won't get that much mRNA being produced, and therefore you won't get much protein being produced. So the affinity of the promoter region for the RNA polymerase II enzyme up here, which is often abbreviated to RNAP2, is crucial basically for determining how much of the gene product of the gene that you actually produce. Okay, right. So what's a transcription factor then? A transcription factor is a molecule, okay, that binds to the promoter regions of genes, and it will have an entire plethora of different genes that it binds to their promoter regions of. Okay, so here is our transcription factor here in orange. Okay, so a transcription factor will bind to the promoter regions of a huge number of different genes. Not all of the genes in the genome, but you know, it's often into the hundreds, okay, that it will bind to. Okay, and what it's going to do is it's going to alter the affinity of that promoter region for binding to RNA polymerase II. Now, at some of the promoter regions that transcription factor binds to, it will increase the affinity of that promoter region for RNA polymerase II, and therefore RNA polymerase II will bind more often to that promoter region, and therefore you'll get more mRNA being produced for the downstream gene of that promoter region, and this increased mRNA will therefore translate into an increased amount of protein being produced. Whereas at other promoter regions, this same transcription factor will bind and decrease the affinity of that promoter region for binding to RNA polymerase II. Therefore, RNA polymerase II will bind there less often, you'll get less transcription of the downstream gene, and therefore you'll get less protein being produced. So at some promoter regions, RNA polymerase II will promote the expression of the downstream gene, and at others it will repress the expression of the downstream gene. Okay, so basically transcription factors change the expression levels of genes, and they do it for a huge number of different genes. Now, ELK1 is going to bind to the promoter region of a very important protein called CPhos, and it's going to increase the expression of that gene. So you're going to get an increase in the production of CPhos. CPhos then binds to another transcription factor, which will be in the nucleus anyway, called CJUN, and they form what's known as a CPhos-CJUN heterodimer. Okay, so it's a dimer because it consists of two proteins. It's a heterodimer because the two proteins that make up the dimer are different. Okay, so let's colour these in. So we've got CPhos I'll have in orange, and CJUN I'll have in blue, I think. Okay, right. Now, CPhos-CJUN heterodimers are transcription factors themselves, and they are pro-cell cycle transcription factors. Okay. In addition, going back a little bit, we discussed that the ERK enzymes, or the MAP kinase enzymes, are going to come into the nucleus and they're going to phosphorylate transcription factors. I've just told you about the phosphorylation of ELK1 and how this is going to lead to the production of these C-phos-C-gen heterodimers down here. Okay, but in addition, the arrival of the ERK/MAP kinase enzyme within the nucleus is also going to phosphorylate another transcription factor, okay, known as MIK. Okay, so here, MIK is a very famous transcription factor. It's often called the cell's most powerful mitogen, which means a molecule that's capable of making the cell divide. Okay, so MIK is very, very powerful. So MIK's going to get phosphorylated and it's going to be activated. Now, the reason I didn't mention this before is now MIK transcription factors, once activated, and C-phos-C-gen heterodimers pretty much do the same thing. They bind to a huge number of different promoter regions of a huge number of different genes, and they change gene expression. Okay, so some genes will have their expression go up, some genes will have their expression go down, and this change in gene expression is going to promote the division of the cell. Okay, so this is what's going to take the cell from the G0 phase into the first growth phase. Okay, and we'll continue this discussion in the next video where we'll discuss the beginning of the first growth phase.